This protocol, for which all of the experimental conditions have been carefully optimized, can be used to facilitate a successful and efficient chemical conjugation of the DEC25 antibody to overantigen. The main advantages of this technique are that it permits a flexible selection of the protein, antigen and antibody, and that it does not rely on genetic fusion of these components.
We have also used this protocol to generate conjugates of hepatitis C virus proteins and anti-DEC205 for in vivo DC targeting. This could likewise be valuable for anti-tumor vaccination approaches.
To start anti-DEC205 production, resuspend a 1 milliliter volume of 1 to 5 million thawed cryopreserved NLDC-145 cells in 9 milliliters of 37 degrees Celsius ISF1 medium, supplemented with 1% penicillin and streptomycin, and seed the cells into a 25 square centimeter cell culture flask for incubation at 37 degrees Celsius and 5% carbon dioxide for 24 to 48 hours. When the cells reach 70% confluency, transfer the entire cell suspension into a 15 milliliter conical tube and collect the cells by centrifugation. Resuspend the pellet in 12 milliliters of fresh, complete 37 degree ISF1 medium and replate the cells in a 75 square centimeter flask. After 48 to 72 hours of culture, split the 70% confluent culture between each of two new 75